Kia ora koutou, everyone, and thank you so much for being here this evening, especially with the weather turning a bit strange out there. My name is Erin Cawston, and I'm joined by Harriet tonight. And today we're going to talk about dementia, what it is, why it matters, and how research here in Aotearoa, New Zealand is helping us stay one step ahead. So this is just a brief overview of what we're going to cover today. We're going to get a quick look at dementia in New Zealand, some of the exciting research that's happening in the Dementia Prevention Research Clinics, and how blood tests might help us detect Alzheimer's disease early. We'll also talk about why funding this research is so important. So dementia is a growing issue in New Zealand and worldwide. In 2020, it's estimated about 70,000 New Zealanders were living with dementia. By 2050, the number is expected to more than double. This has a huge impact, not just financially, as we can see here, and this is only the estimated financial burden, but emotionally and socially for our families, friends and communities. So what is dementia? Dementia isn't just one disease. It's a term of a group of conditions that affect the brain. It causes problems in memory and thinking and daily life. It's progressive, meaning it gets worse over time, and it affects everybody differently. So, as I said, dementia is an umbrella term. Alzheimer's disease is the most common type of dementia. It's making up approximately 55%. And there are others too. There's things like vascular dementia and dementia with Lewy bodies and others. Today we're going to focus about Alzheimer's disease in particular. So forgetfulness is a normal part of aging, but Alzheimer's is different. It's not just forgetting where you put your keys, it's forgetting where you live. So it's about the magnitude of change in memory. It's really important to note Alzheimer's and dementia is not a normal part of getting older. So this is just a basic overview of what we know about Alzheimer's disease, but there's a lot we still don't know, and that's important to remember. Alzheimer's is caused by harmful proteins building up in the brain. These proteins that we know about are called amyloid and tau, and they disrupt how the brain communicates and eventually lead to changes in brain cell death. So this is a brain that is slowly shrunk. This is a neuron, which is one of the most important cells in the brain, and these are what we call amyloid plaques, so the amyloid build-up. And these are the tau tangles that occur within the neuron. And eventually what happens is this causes the neuron to break down, leading to the cell death and this shrinking of the brain. That's what causes the common Alzheimer's disease symptoms we see as such as memory loss and difficulties thinking and functioning in everyday life. So right now, diagnosing Alzheimer's disease is tricky. We rely on symptoms and rule out other causes. Brain scans and spinal fluid tests can help, but these are expensive and not widely available in New Zealand. This is where something like a blood test could provide a cheap, accessible and equitable solution for aiding clinicians in the diagnosis and tracking of the disease progression of Alzheimer's disease. So this diagram looks a little bit intimidating, but I will go through it and explain it, but Alzheimer's disease doesn't happen overnight. Changes in the brain are estimated to start approximately 20 years before symptoms appear. That's why we're focused on the earliest signs that we might be able to intervene. So this is what we call a model of the continuum of Alzheimer's disease. We see this preclinical stage here. That's before symptoms start to appear. And it is before this MCI, which is mild cognitive impairment. So mild cognitive impairment is a potential precursor to Alzheimer's disease. Mild cognitive impairment is a condition in which an individual might see some changes in their thinking abilities that are noticeable to maybe the person that's affected and their family members and friends, but do not affect their daily lives or ability to carry out everyday activities. It's a really important thing to note that this is a hypothetical model for the continuum of AD. Not everybody who has MCI will go on and develop Alzheimer's disease. Some people with MCI are at greater risk of AD, and in some individuals, mild cognitive impairment reverts back to normal cognition or remains stable. While there's a lot of research on Alzheimer's disease, there is still much we don't understand. And for research to be truly useful in New Zealand, it's important that studies include a New Zealand context, because it's our unique genetics and environment that may influence how this disease progresses. Therefore, identifying which individuals in this early stage preceding Alzheimer's disease that go on and develop Alzheimer's disease versus those that don't is a really important aspect and one of the main drivers for establishing the New Zealand Dementia Prevention Research Clinics, which I'm going to talk about now. So in 2016 we began the New Zealand Dementia Prevention Research Clinics, and yes, that's a mouthful, but what it is at the heart is a multi-centre, multidisciplinary, longitudinal observational study. It's a lot of words. But our main goal is to improve the understanding of AD and dementia using a team of experts in the fields to develop interventions and delay or prevent the progression to dementia. This study comprises of research centres in Auckland, Christchurch and Dunedin and involves participants from the community. We aim to recruit people and follow them over time, and it involves people who are cognitively healthy, older adults, people who notice memory changes but when they get testing they're normal, and we call these subjective cognitive decline, and people with mild cognitive impairment but are still functioning okay in daily life. And then there's people with Alzheimer's disease. So participants are followed every 2 years. They come in and see our clinics, so we can track who stays healthy and who develops dementia, and most importantly, why. So one of the major aims of the research is to identify biomarkers or biomarker signatures that predict which individuals with this mild cognitive impairment, the precursor to Alzheimer's disease, will progress to dementia in a New Zealand sample, and to identify factors that might influence this progression. And to do this, we're looking at a raft of different things. We also wish to understand the experience of individuals and their whānau living with mild cognitive impairment and dementia to help better understand this disease as well and how it affects the wider community. We want to investigate and identify mechanisms underlying the development of these conditions and to test a range of interventions when they become available, such as novel compounds, cognitive, physical, sensory, lifestyle, social, all sorts of different things that might prevent or delay progression. So this is just a snapshot of where we are at the moment. To date, over the last 10 years, we've assessed 550 participants nationally. We currently have 431 people enrolled with individual classifications into groups across the AD continuum. So like I showed you that diagram before, we have controls, people with Alzheimer's, people with mild cognitive impairment at various stages, and then subjective cognitive decline as well as, and for our participants, we see them, as I said, every 2 years, and we collect a huge range of data, clinical assessments, brain scans, lifestyle information, blood samples, and we're going to hear more about those today. This helps us build a complete picture of the person's health, brain health, and general health at each visit. So I'm just going to run through a little bit of an idea of what we assess in the clinics and how that helps us. We have an extensive clinical arm with neurologists, clinicians, neuropsychologists, and research nurses. And this is just a brief overview of some of the data we collect in the clinical arm. We also have an extensive neuroimaging platform, so scanning of the brain. The brain scans that occur every 2 years take 45 minutes and comprise of both clinical sequences or clinical reads as well as research sequences. So looking at new and novel things that we might be able to see in a brain scan. Additionally, we're now carrying out what is called an amyloid PET scan or an amyloid brain scan. So, I talked about amyloid in the brain, so we can scan that. But as I said before, they're expensive things and not widely available. So what we want to do is measure amyloid brain scans in some of our participants and controls as well. This is the neuroimaging slide. And then we also have a blood platform, and I'm very proud to be the National Co-Director of the Dementia Prevention Research Clinic's blood platform. When I joined in 2016, we were in the setup phases, so I was put in charge of basically setting this whole thing up. Because I have a background in what is called medical laboratory science or medical diagnostics. So I also lead research projects utilising our clinical samples from the DPRC, and Harriet and I are going to give you a snapshot of some of the research we're currently doing. So, how could a blood test be useful to a clinician, and how can blood tell us anything about the brain? So certain proteins are made in the brain that, like those involved, such as amyloid and tau that I talked about before, and these build up. These proteins can enter the bloodstream, but they're often at very low levels. Historically, we have not been able to measure these proteins because they're at such low levels. But in the last 10 years, we've developed new technologies called ultra-sensitive technologies, such as the machine shown here, which we now have the only one in New Zealand in our lab. And this is called a single molecule array, which has the ability to measure levels down to a single molecule at very low concentrations. So measuring these biomarkers can give us insight into how the disease progresses, support diagnosis and track progression. Before they can be used in clinic, however, we need to determine which biomarkers are robust and what information they can provide. This is where our research comes in. So today I'm going to tell you about a piece of work that I have done comparing 9 promising blood biomarkers between those different clinical groupings that I've described for the clinics before, from 253 participants in our study. The blood samples from these participants span that AD continuum from healthy older adults through to probable Alzheimer's disease. And we've used that piece of technology from the previous slide, and this is called a SIMOA, or single molecule array. So this diagram shows a brain cell, like I showed before. And this is a neuron, and this is what we call an astrocyte. So it's another important brain cell. So we can see here that we've got those amyloid plaques and those tau tangles in here. And so these are 9 different markers that I've listed on here that we've measured in the blood samples from our Dementia Prevention Research Clinics. Another thing to note is when the neuron breaks down, we can also measure these things that describe that the cell is dying as well, like neurofilament light. We can measure this neuroinflammatory marker from astrocytes called glial fibrillary associated protein, or we call it GFAP. And then we have the amyloid proteins, as well as these phosphorylated tau proteins. And we see these 217, 181, 231. And those numbers really just mean where the tau protein has been modified. So these are the 9 markers that we've measured in our blood samples. So I'm not going to show you all the data of course there was lots of data and we had it for all of those different stages, but really I wanted to highlight these two particular proteins. So these graphs show that blood biomarker levels from p-tau217, so one of the markers I showed before, and GFAP across the clinical groupings, from the cognitively unimpaired here to Alzheimer's disease. And this grouping here is non-amnestic MCI. So these are people that are having cognitive impairment, but it's not related to Alzheimer's disease via clinical classification. So what we see here is between the controls, or cognitively unimpaired, and Alzheimer's disease, for phosphorylated tau and GFAP, we see this significant increase in the protein levels. And we see the same, but not to the largest extent, in GFAP. So this shows that as the AD continuum progresses, we can get increases in these proteins. Additionally, we can see here between the AD and non-amnestic AD, so people who have memory complaints but it's not Alzheimer's, we can also see that there's a higher level in the AD versus there is in the non-amnestic MCIs, which means we can distinguish between people with Alzheimer's disease and other types of dementia. So, don't worry about all of the parts of this table, but basically this is a ranking from the differences between the groups. And what we see here is that p-tau217 and GFAP have the largest differences between those different groupings of clinical classifications, with p-tau217 actually having a really big difference. So the take-home message here from this study is that we've found in the DPRC study, or Dementia Prevention Research Clinics, that certain proteins found in the blood, especially p-tau217, are good at identifying Alzheimer's disease, which means that p-tau217 may hold promise to help clinicians and to diagnose AD. Part of Harriet's PhD is to look at some of the most promising proteins that I've described here in our DPRC participants and see how these change over time, with the hope that we might be able to get more insight on the earlier stages of the disease and the progression over time. So what's next? As we saw in the DPRC samples, we saw this significant increase in this p-tau217. At the same time, internationally, other groups were actually looking at this biomarker as well, and they found the same. And so I don't know if any of you people have seen this, but this hit the Alzheimer's disease field internationally as a biomarker for AD. And the field was blowing up with excitement about this new biomarker called p-tau217. So why is this p-tau217 such big news? Well, as I said before, it's strongly associated with AD-related amyloid and tau build-up in the brain. It excels at differentiating people who have AD over other dementias and at detecting AD pathology in MCI, or that mild cognitive impairment stage. The test also shows the largest changes in the disease, and these things make it a promising biomarker. I should emphasise, though, that p-tau217 at this stage will not be used if it comes into a diagnostic platform as a standalone diagnostic test, but rather aid a clinician in their overall clinical workup. So as I said before, p-tau217 shows a strong correlation with the build-up of amyloid in the brain, which we can measure using those costly amyloid scans. So considering our previous study in the DPRCs, we are working towards how can we use that blood test for p-tau217 to get more information about the levels of amyloid in the brain. In a pilot sample of 63 participants who had blood samples and brain scans at the same time, we measured p-tau217. And these are the people that tested positive for the amyloid via that brain scan, and these are the people who tested negative. And what we can see is, looking at the p-tau217 in blood, we can see that these individuals had a much higher level of p-tau217 in the blood if they had a positive amyloid scan. So giving us confidence that this p-tau217 can detect the amyloid in the brain. And these people with negative had low levels. So internationally, where the field is moving in this area is to create these thresholds, because it's not necessarily a positive or negative read when it comes to amyloid. So internationally, people are working towards saying at a certain level, above that level, a person is high risk of being amyloid positive if they have this blood test. And below a certain level, they're at low risk of having an amyloid. And that's important, because if the clinicians have this information, then they don't need to send them for costly PET scans. They can use this blood test. But it's also important that there will be a zone in this area where it's neither high risk or low risk, it's intermediate risk. And these people are the people that potentially need to have the costly brain scan, or that need to be followed over time. And so this really helps the clinician to sort of work out at what stage they need to start doing some of these tests. So building on the pilot data from the previous study and the growing momentum in the AD field, at the beginning of last year, we applied for a Neurological Foundation grant to further this work. We were fortunately successful, and therefore this grant will enable us to look at a larger number of people in the DPRC study to build on this New Zealand first work. We are working together to progress the New Zealand health system in this area. This new study that we have funded will allow us to increase our number of people to 250 who have had both brain scans and blood samples. As part of Harriet's PhD, she will measure the blood p-tau217 by the analyzer I showed you before and help derive that 3-range cut point that I described before, but in a New Zealand context, because we know we may have unique features that might change those levels. Importantly, we wish to use DPRC data, or Dementia Prevention Research Clinic data, to inform New Zealand blood biomarker diagnostic processes in New Zealand, and to aid their implementation in clinical assessment in a New Zealand context. So what else can we do to aid this clinical implementation of blood p-tau217 in New Zealand? There's still a lot of work to do here. We need to investigate how p-tau217 changes over time, and as I mentioned, Harriet will be doing some of this work in her PhD. We're also using DPRC data to inform health policy in New Zealand, and how this might enable implementation into the clinical workflow. We also want to engage with health economists to use this data that we've gained from our studies to show the economic benefit of bringing this test into New Zealand. But there's also what platforms can we use to do this testing that would be useful in a New Zealand clinical lab. So future directions. We have lots of work to do looking at the longitudinal blood biomarkers for the DPRC participants. We want to specifically look at more depth over time into that MCI group, or mild cognitive impairment group, that I talked about, and how blood biomarkers might be able to differentiate between those who progress to AD and those who don't. And while AD is defined pathologically by that amyloid and tau in the brain, we know AD is complex and involves multiple molecular pathways, or different pathways and interactions that cause the disease. Therefore, we still have more work to do to look further into these pathways and look at other blood biomarkers for AD. So that is where I now hand over to my excellent PhD student, Harriet, who will give you a snapshot of other work we're doing looking at these other pathways and studies that I've mentioned. Thank you.
Awesome. Thank you for that introduction, Erin. So as Erin said, Alzheimer's is a very complex disease. So Erin's been talking to you about amyloid and tau, but there are other pathways involved. And as part of my PhD, I wanted to explore some of these a little bit further. So I'll be talking to you about inflammation and vascular damage in Alzheimer's disease. So firstly, in the brain, blood vessels are surrounded by lots of other cells. So some of those cells like astrocytes, which Erin has been talking about. And the cells surrounding the blood vessel itself, and I've got a close-up down here. So these cells here form what is called the blood-brain barrier. So this is essentially a shield that lines the blood vessels, and it's a protective shield which normally keeps harmful substances in the blood from getting into the brain. But in Alzheimer's, this shield can become leaky or damaged, and things from the blood can leak into the brain, and this can trigger inflammation. So inflammation is the body's natural response to injury or infection. It's how your body fights off harm. But in Alzheimer's, this response can become chronic or long-lasting. So this can actually end up damaging brain cells further, especially if a brain is already vulnerable from the amyloid and tau buildup that Erin has been talking to you about. So part of my work is trying to measure molecules related to this inflammation in the blood of our DPRC participants and see if there are any differences in individuals at different stages of the disease. So I'll just explain this diagram that I've got up here. So a pilot study that I conducted, so this had 10 people, so 10 controls and 10 people with Alzheimer's. And I was screening for lots of different molecules in the blood. So down here, I've just got a list of 105 different molecules that I was looking at. And down here are all the different participants. So each column here is a different person. And the colors just mean, so if it's dark blue, it means that there's a lot of the protein present. And if it's white, it means that protein probably isn't present at all or at very low amounts. So just looking at this, it's kind of hard to see any patterns. We can see that there's a lot of differences between people, but not easily to see between the groups. However, on further inspection, I was able to see three proteins with significant differences between the groups. So here, these are chitinase 3-like-1, IGF-BP2, and osteopontin. And all three of these molecules are involved somehow in the inflammation pathway. But they also have lots of different roles in the brain and throughout the body as well. So these three candidate proteins, I aim to investigate these further in a larger sample size. So before I only had 10 participants in each group. Now I want to do it with 35 participants and also add in more groups. So I was just looking at the ends of that continuum that Erin was talking about, but now we want to look at some of those earlier stages as well. And I also want to use a more sensitive method. So the method of that diagram that I showed you before, it can just show us if there's an increase or a decrease, but it can't tell us the exact amount in the blood. So now I want to use a more sensitive method, like that machine that Erin showed you, which can actually tell us more accurate measurements. Cool, so alongside our blood work, we've got some pretty pictures of some human brain cells up here. So I'm also using a cell culture model to investigate how one of the key cell types in that blood-brain barrier that I was telling you about. So in this diagram here, these cells are these orange ones here, and these are called pericytes. So these line the blood vessels, and they're found not just in the brain, but the ones that I'm looking at here are brain-derived. So we've gotten these from human brains which have been very generously donated to the Neurological Brain Bank and also the Hugh Green Biobank. And I'll just explain a little bit about what these images actually are. So I've stained different parts of the cell in different colors. So in blue here, so all of these blue dots in each image they all just show a different cell. So in blue, I've stained the cell nucleus, which houses the cell's DNA. So every cell has one of these. So each blue dot is a different cell. And in green, you can see here, I've stained a protein called ICAM1, which we know to increase in inflammatory conditions. And in red and pink here, I've stained for one of those proteins that I saw to increase in the blood. So this is IGF-BP2, which is produced by pericytes. And then this one looks a little bit different here, but that's because I've stained for a different protein, which is a structural protein of these cells here in blue. So some of the work that I am doing with these cells, so I'm comparing the inflammatory response of these cells, and we have cells from people who've donated their brains, from cognitively healthy older adults and people with Alzheimer's disease. So I'm comparing if there is a difference between these cells in how they respond to inflammation. And we also have their genotypes. So ApoE4 is a gene which you may have heard about in the news a little bit. This gene does increase the risk of having Alzheimer's. It doesn't guarantee it, but it does increase the risk. So with the cells, we have some of the cells have this gene and some of them don't, or this type of gene. And I want to see if that has an influence in how they respond to inflammation. Cool. So as both me and Erin have been talking about, there is still a lot that we don't understand about Alzheimer's, and there is a lot of work in New Zealand going on to help us understand what is going on in the brain. And that is why continued research, like what we're doing at the DPRC, is so important. And why funding like that from the Neurological Foundation is essential to make this possible. Brain research is expensive. I mean, the DPRC relies on advanced equipment, blood analysis, brain scans, and years of follow-up, and this all costs a lot of money. The DPRC has already built an incredible foundation. As Erin said, we have hundreds of participants and thousands of clinic visits. And we are now in a critical phase, ready to continue this work and use the data that we've collected so far to start answering some of these really important questions and unlock the full potential of this national resource. Funding is essential to continue tracking these participants over time, support biomarker discovery, preparing for new treatments on the horizon, and to develop tools tailored to New Zealanders, not just based on overseas data. And we are greatly appreciative of all who have funded us. We can't do this without you. So I just want to finish by thanking everyone who supports and participates in this work, including the DPRC participants and their families and friends, their donors and their families, my supervisors, Dr Erin Cawston, Professor Mike Dragoner, and Professor Lynette Tippett, the Centre for Brain Research, including the Hugh Green Biobank team, and Dr Rebecca Johnson for all of their help with that human brain cell work. I also want to acknowledge the DPRC team, led by Professor Lynette Tippett. And this team consists now of too many people to fit on this slide, but every one of them does such an amazing work to keep this study up and running. And I also want to acknowledge the Auckland DPRC Blood Biobank and our lab techs Celestine and Estelle, and our collaborators in Sweden, including Professor Henrik Zetterberg. And of course, a huge thank you to all of our funders, and with a special shout out for the Neurological Foundation for funding my PhD scholarship. And thank you all for being here. Thank you so much for your time.